Now we can go into the inner ear, which is located deep in the temporal bone, and you want to be thinking of kind of a hollowed out portion deep inside your skull, about an inch and a quarter or so behind each of your eyeballs. And that is where the inner ear and its two sensory divisions, balance and hearing organs, are located. The vestibular division, I'll just mention first because we'll just kind of bring it up and then leave it behind. But the vestibular division of the inner ear is what is responsible for transducing or for converting to nerve impulses the fluid motion inside your skull that is associated with the movement of your head through space. So it helps you maintain your posture, helps you maintain specifically an upright posture, and it also detects the movement of your head as you accelerate either in a straight line, forward and back, up and down at an angle, or even in a rotational direction. And if any of you have ever had any problems with vertigo or dizziness, you understand that disorder of the vestibular system is more debilitating, probably in the long run, than hearing loss. And so one of the nice things about our audiology program, if I can plug it here for just a moment, is that we have an excellent vestibular program, an excellent sequence of courses in the vestibular sciences that really sets our program apart from many others because they are taught by a specialist at the VA named Dr. Faith Aiken, who does excellent research along with a colleague of ours, Dr. Owen Murnane, in this area. And again, I just bring it up as kind of a plug for the program, but it is very interesting, some of the research they do, and of course, they're always looking for subjects. So if any of you are interested in participating, that would always be an available option for you, although it would mean you'd probably have to go to the VA for it. We'll, of course, focus our attention on the auditory division of the inner ear, the so-called cochlea, which is the organ of hearing. And if we look inside the skull, we can see really two areas here. First of all, let's get oriented before I start putting those circles up there. Here's the eardrum. So the ear canal would be out here. We talked about that last time. Here are those middle ear ossicles. Again, we have discussed these already. The malleus, the incus, and the stapes, or the hammer, the anvil, and the stirrup. You can really see how the stapes bone resembles a stirrup here. We have a round window over there, and the oval window would be right here where the stapes foot plate fits into this opening in the skull that is filled with fluids. The fluids are made up of different ionic concentrations. More about that in just a little while, but just recognize that all of this brown shaded area is bone. It's the temporal bone of your skull, and inside it, again, are these cavities, these labyrinths, as they're called, because they are kind of twisty and turny. And these labyrinths are filled with fluids. So you've got these very intricately shaped cavities filled with fluid, hollowed out of the temporal bone in your skull. And it will be inside these fluids that the transduction process for both balance and hearing take place. Here are the balance organs, and you see they consist of the semicircular canals, the utricle, and the saccule. And then the auditory mechanism is housed in this coiled up snail shell shaped structure called the cochlea. And we will look more closely at the cochlea, but just recognize again that movement of the eardrum vibrates the ossicular chain. The movement of the ossicles will, in and out of this space, will set these fluids, both the white and the blue fluids, into motion, and in doing so generate either balance-related sensations, auditory sensations, or in some cases even both. Frank Netter has now kind of dissected the whole thing out for us, and here we see the auditory nerve, the eighth cranial nerve, or the vestibulocochlear nerve, which actually has these two divisions, one of which goes to the cochlea, and the other of which serves the balance organs. Here's the cochlea, and it would be coiling away from us as we're viewing at it from or looking at it from this angle. And you can see how that nerve trunk kind of disappears up into the coils of the cochlea. More on that in just a moment. Right here, that would be where the nerve is kind of disappearing up into the coils of the cochlea. So let's take a step back now for just a moment. And imagine that we have sliced this organ in half like this, and we're going to open it up and look inside. This is what it would look like. So this part right here, this so-called apex or helicotrema, is exactly the same as this location right here. So again, right here, the tip top, the apex of the cochlea would be this location right here. Now, if looking at it in this photograph, you would think that this was the bottom and this was the top. In fact, the basal region down here is actually the most posterior section, and the helicotrema, this part right here, is the most anterior. So the thing actually kind of coils out, coils forward in your skull, kind of toward your eyeball. So this location right here is actually anterior, right there, and this would be the most posterior section. With respect to that nerve, here are the nerve fibers going up in through the base of the cochlea into those coils. And here again it comes in through the base, and it's going to synapse onto, the nerve fibers are going to synapse onto hair cells that are located inside the organ itself. What we see from this image is that the fluids of the cochlea actually are separated from one another by these membranes. And let me skip ahead for just a moment. Here are the membranes that separate the fluids. You have one kind of fluid up here, the same kind of fluid in here. Both of those cavities are filled with what's known as perilymph. And then you have endolymph in this middle cavity there. Going back to this image, all the white stuff would be perilymph. All the blue stuff would be endolymph. Perilymph, perilymph, endolymph. Perilymph, perilymph, and endolymph. We'll talk more about those fluids in just a little bit, but just recognize that's how the cochlea is arranged with respect to the organ of hearing, the organ of Corti right here, or right here, and then the membranes that separate the fluids. Now moving back one slide, you can see from this dissection that the cochlea does have this kind of coiled up snail shell shaped appearance. And in this image here, what you can see is that there actually are these layers of bone that support the structures of the cochlea, almost like a corkscrew. And so the layer of bone, which emanates to about right here, again, is the resting place, or upon it sit these different cavities filled with fluid inside the cochlea. And the whole thing revolves around this center axis called the medialis, right there, spelled modiolus, but pronounced medialis. And the whole thing kind of rotates around that the way that the ridges on a corkscrew revolve around that center of the corkscrew, or the way that a spiral staircase revolves around that center pole, I guess, that holds the thing in place. Looking over here, we see the oval window, which we spent quite a bit of time discussing last time. That's where the stapes footplate would fit in. And then here's that round window, which again is just kind of across the way, if we look at it here, just adjacent to the oval window, and we'll see why that's important later on when we look at the function of the cochlea. So again, let's back up. We've got these two divisions, balance and hearing. The auditory or the vestibulocochlear nerve, the eighth cranial nerve, sends fibers into both organs. The fibers travel through this trunk up to all the different areas of the cochlea. They kind of branch off, almost like spokes on a bicycle wheel. You can kind of see that here, almost like spokes on a bicycle wheel to all the different locations in the cochlea. And remember, or maybe not remember, but we're going to get to the idea that the cochlea is going to process different frequencies of sound in unique and different ways. And we'll see how these fibers kind of serve that function. So again, here they are coming through the base of the cochlea, and we'll see how they synapse onto hair cells, onto sensory mechanisms throughout the length of the cochlea. And again, we've got the rotation around the center axis there. This is a nice image that kind of summarizes a bunch of that. Let's now name the different cavities in the cochlea. This one, sort of what appears to be on top, is the so-called scala vestibuli. The one on the bottom is the scala tympani, and the one in the middle is the scala media. If we jump back here now, we'd see scala tympani, scala media, scala vestibuli, scala tympani, scala media, scala vestibuli, all the way throughout the organ. Now, the reason they're named that way has to do with this diagram here. So let me get rid of these circles. So you can see here again, scala vestibuli kind of appears to be on top. The cochlear duct is the scala media, and then the scala tympani is down here on the bottom. I want you to notice this relatively large open area right here. This is the so-called vestibule of the inner ear. And you'll notice how the stapes footplate fits into the oval window at the vestibule. If you now follow the vestibule up into the coils of the cochlea, right through here, you see that you are in the scala vestibuli. So the scala vestibuli actually opens up into the vestibule. On the other hand, the scala tympani, which is this cavity over here, opens up into the middle ear space, which we've already identified as the tympanic cavity or the tympanum. So we're going to call this area, this cavity right here, the scala tympani, because it opens up through the round window, or is separated by the round window from the tympanum. And then this scala vestibuli opens up into the vestibule of the inner ear. And that's how those two cavities get their name. Believe it or not, the scala media is in between the two, so it's in the middle, or kind of medium, I guess you could think of it. And surprise, surprise, we're actually going to call it something that makes some intuitive sense, the scala media. So there we are again. You see also here the spiral ganglion. The spiral ganglion is a collection of nerve cell bodies that are the cell bodies for the neurons that actually synapse onto or communicate with the sensory cells in the organ of Corti. The scala vestibuli is separated from the scala media by Reisner's membrane. The scala tympani is separated from the scala media by the basilar membrane. And again, we have different types of fluid in the different cavities. We have perilymph, which is a lot like ocean water, in the scala vestibuli and scala tympani. And then we have endolymph in the scala media. And again, we'll talk more about those in just a little bit. This so-called stria vascularis provides nutrients and the blood supply for much of the cochlea's structures, and also maintains the ionic concentration of the endolymph. And the spiral ligament, just as any other ligament, is connective tissue, kind of holds things in place. Here's a close-up of the organ of Corti. So again, we're going to now take, what we've just done is we've taken one of these coils and we've magnified it, and now we're going to take this area right here and magnify it, and we see that the organ of Corti consists of a few pieces of tissue and then a variety of different cell types. The cell types that we're most interested in are the so-called inner hair cells, IHC, inner hair cells, and outer hair cells, or OHC. Okay? And these different hair cell populations are actually the sensory elements that make the cochlea or that enable the cochlea to perform its primary function, which is to transform sound energy into nerve energy. And you see that there's a very dense supply of nerve fibers, that synapse onto the inner hair cells. There are also nerve fibers that synapse onto the outer hair cells, but it looks like there's many more that go to the inner hair cells, and that's for a good reason. That's actually the way that it is. The significance of that is that the inner hair cells turn out to be more important for the transduction, the transformation of sound energy into nerve impulses than the outer hair cells. The outer hair cells have a different job. And the main objective for the outer hair cells is to help increase the sensitivity of the inner hair cells. And when we look more closely at the hair cells in just a few moments, we'll see evidence of that. You've got a membrane down here, the so-called basilar membrane, and you've got another membrane up here, the so-called tectorial membrane. Try not to get confused by TM, which we've also used as an abbreviation for the tympanic membrane. This is a very different type of membrane made up of different kinds of tissue. It behaves very differently. The only thing similar about it to the tympanic membrane is its abbreviation, so try not to get confused there. When we're talking about the inner ear and you see the abbreviation TM, the odds are that it means tectorial membrane. Now, here we are again looking at the cochlea. Here we're kind of looking at it from a different side, though, if you will. So on this one, the inner hair cells are on the left, the outer hair cells are on the right. In this one, the outer hair cells are on the left, the inner hair cells are on the right. And this should remind us that this organ actually coils around. So over here, the outer hair cells would be facing away from the modiolus. And over here, the inner hair cells would be facing toward it. Over here, the outer hair cells would be over here, and the inner hair cells would be over here, right? It's because the thing kind of coils around like that. So we see here, again, the tectorial membrane kind of sitting across the top of the organ of Corti, and these outer hair cell little hairs, as they're called, or stereocilia, project up into the tectorial membrane, and that has some implications for the processing of mechanical vibration or sound energy. The spiral limbus is this area over here, and it forms the attachment for the tectorial membrane toward the modiolus portion of the organ. So again, if we went back up here, we would see the spiral limbus would be over here. And if we go back up here, here's the spiral limbus right here, forming that attachment point for the tectorial membrane and some of the other, supporting some of the other structures. Limbus, by the way, is just a margin or a border or an edge. That's all that means. I like this image because it gives us that three-dimensional aspect that we've been lacking in the other images. You can see over here you've got one inner hair cell and three outer hair cells in there. You've got the spiral limbus in here supporting and forming an attachment for the tectorial membrane, which is right here. And you also see these nerve fibers, thousands and thousands of nerve fibers that synapse onto the hair cells, exiting through the bone of the skull into what would be the spiral ganglion somewhere in here, and ultimately the eighth cranial nerve. So we see the bony capsule of the organ here, and then we see this membranous area that contains the fluids that fill the scala vestibuli, the scala tympani, and then in here the scala media. The hair cell populations are quite different from one another. There are, at birth in humans, probably somewhere between 15 and 16,000 total hair cells. There are about 3 times as many outer hair cells as inner hair cells. So when we look at something like this and we see 3 outers for every inner, that's not an accident. That is actually the way that it is arranged. And these two populations of hair cells are quite different from one another, and they do serve different functions. We believe that the vast majority of sound transduction, that is to say transformation of sound into nerve impulses, is carried out by the inner hair cells. And the reason for that is that when you count the nerve fibers that actually attach to these hair cells, it turns out that there are about, out of every 20 fibers, 19 of them synapse onto inner hair cells. That means that 19 out of 20 fibers in the auditory portion of the eighth nerve synapse onto the inner hair cells. This makes us think that the inner hair cells are the so-called primary transducers of sound in the auditory system. The outer hair cells actually have some interesting mechanical properties that we'll look at in the next lecture. And the result of those mechanical actions carried out by the outer hair cells seem to be directed at improving or increasing the sensitivity of the inner hair cells. So just to kind of jump ahead for a moment, if you lose all your inner hair cells, you will be profoundly hearing impaired. If you lose all your outer hair cells, you'll have probably about a moderate hearing loss at worst, as long as the inner hair cells are still okay. So these are the primary transformers of sound energy into nerve impulses. These seem to make the inner hair cells more sensitive. If you look at a photograph of them with the tectorial membrane removed, this is a pretty decent image of what they would look like. Again, you got 3 rows of outer hair cells for the 1 row of inner hair cells, or if you want to count them, for every 1 inner hair cell, you've got 3 outers, and on like that. These are a couple of different sites for animations and different kinds of images that should help you kind of picture what might be going on inside the cochlea. You know, the cochlea, because it's encased in bone, we can skip back here and kind of see that all this is bone out here. If you want to observe it, you got to actually break it, because as soon as you break through the bone to get to the cochlea, the fluids will start leaking out. So it's very difficult to examine directly. This, of course, would be a photograph taken from a cadaver of some animal, probably a chinchilla. And, you know, the idea is that it's very difficult to get at this organ, but once you do, and if you can strip away some of the supporting membranes and stuff, you actually can see these sensory cells, and they really are quite remarkable. You want to consider the fact that you've got all of these sensory cells located inside that cochlea, which again is about the size of a box whose sides are the size of a dime. It's about the size of your fingertip. Now, These little white projections here are the so-called stereocilia, or the hairs that project out of the hair cells. They're not actually hairs, but they look enough like hairs so that the sensory cells have, over the years, acquired that name. We're not going to focus too much on what these projections actually are. Let's just recognize that they are called the stereocilia of the inner ear. And it is through their action that the transformation of sound energy into nerve energy is going to take place. Okay, so again, I encourage you to take a look at some of these different images from that old internet, and I think you'll find that they are going to help you picture what might be going on inside the cochlea. These structures are so small that the only way to photograph them is by using a scanning electron microscope. And so here is a scanning electron microscope image of the cochlea, kind of similar to maybe that image there, except that this is actually a photograph, and you can see the inner hair cells going off into the distance there. You can see 3 rows of outer hair cells right there. So the thing would have to kind of coil this way, right? Because there's the inner hair cells. The medialus would be over here somewhere. And you can see actually some of the outer hair cells themselves. Can't really see an inner hair cell very well there, but you can see also these nerve fibers that have to cross through the organ of Corti in order to get to the outer hair cells. Because remember, the nerve fibers are going to travel through this trunk, if you will, that is kind of oriented near the medialus of the cochlea. Again, this is a microscopic image. We see dozens of sensory cells here. Keep in mind that inside your inner ear you would have somewhere around 15,000 of them. If we look at the hair cells at the apex of the cochlea, or the organ of Corti, in a slice through the apex of the cochlea versus the base of the cochlea, you can see how at the apex the outer hair cells seem to be a little bit stretched out, a little bit longer, perhaps a bit more massive, which would make sense, as we'll see in a few minutes. The apex of the cochlea, near the helicotrema, is where low-frequency sounds are processed, and the base of the cochlea, near the stapes footplate, if you will, is where the high frequencies are processed. As I said before, the different hair cell populations are physically distinct from one another. You see with respect to the inner hair cells, they are this kind of flask-shaped, floppy structure with a very rich supply of nerve fibers synapsed on to the hair cell itself. And again, 19 out of 20 afferent or sensory fibers will synapse onto the inner hair cells. With respect to the outer hair cells, you see how there is a kind of a very rigid support structure for the outer hair cells. You see how the outer hair cells have this very rigid flask-like, or tube-like shape to them, cylindrical. And you also see far fewer fibers synapsed onto the outer hair cell when compared to the inner hair cell. So again, the higher number of these afferent fibers suggests that as far as converting sound energy into nerve energy, the inner hair cells are going to be more important because they're informing, if you will, more fibers of their being stimulated, or of the sound that's being presented, whereas the outer hair cells are going to be responsible for actually making it easier for a sound to stimulate the inner hair cells. And when I say make it easier for a sound, what I mean is a sound at a level that would otherwise not be detectable will be able to be detected because of the action of the outer hair cells. That's why when you lose the outer hair cells, you really only lose the ability to hear the lowest sound levels that are associated with a input level that's even below that of typical speech. And that's why when you lose the outer hair cells, all you lose is the ability to detect those low-level sounds. Hence, you get a mild to moderate hearing loss. If you lose those inner hair cells, though, you lose the ability to transform sound into nerve impulses over a wide range of levels, and that would result in profound deafness. Now, how is it that the inner ear does its job? Well, we're going to have to look more closely at the hair cells later. Let's introduce this idea, though, by looking at what the basilar membrane of the cochlea is made up of. So the basilar membrane is composed primarily of connective tissue, and it turns out that it is of different amounts of stiffness from place to place. So let me back up here. Here's the basilar membrane right here. Here it is right here. If we go up here, the basilar membrane would be this thing right here, right? So it is the area upon which, or the membrane upon which, the organ of Corti, the hair cells and the hearing organ, sit. It's kind of the sensory surface for your inner ear. Well, it turns out that the membrane is very narrow and stiff at its base, very wide and relatively floppy at its apex. Now, we've already talked about the relation between stiffness and resonance frequency. So you should be able to predict that the higher frequencies will be processed in the region of the basilar membrane where it is stiffer, and we would imagine then at the area where it is floppier that lower frequencies would be processed there because the membrane is less stiff. So remember, resonance frequency is proportional to stiffness. If I've got an object that is composed like the basilar membrane of different areas that have different amounts of stiffness, the highest frequency sounds will be processed at the stiffest location. The lowest frequency sounds will be processed at the least stiff location. What sets the basilar membrane into motion? What stimulates the organ of Corti, the organ of hearing? That would be the motion of the cochlear fluids that is linked to the vibration of the stapes bone. So it's almost like we've got I'm going to scoot back here real quick. It's almost like we've got a piston that's going to vibrate in and out of this area, and in doing so, send waves of pressure, send a pressure wave through this fluid-filled cavity. Now this fluid is compressed, it cannot be expanded, so when you push on it over here, it's going to bulge out somewhere else. It turns out that it bulges out at the round window, and again we'll talk more about that next time, but recognize for now that this system is a fluid-filled mechanical system that will be stimulated by, set in motion by, activated by the movement of the stapes footplate in and out of the oval window. So if we now go back to here, we recognize that this system is a hydraulic system with a piston on one end and structures on the other end. The piston is the stapes footplate. The structures that are going to get moved are the hair cells, the sensory cells in the organ of Corti. Now, the basilar membrane's tonotopic organization should remind you of things that you're more familiar with, things like xylophones or pianos. And if you picture a piano in your mind, you know that when you hit it with your left hand, generally the tones that come out will be lower in pitch than when you hit it with your right hand. And if you can get your hands on a piano that you can lift up lift the hood up, if you will, so that you can actually look at the strings. You can't do this with an electric piano, but you can with an upright or an acoustic or a grand piano. You can see that the strings that correspond to the keys on the left side of the piano are much thicker than the strings on the right side. Kind of like if you picture a guitar in your mind, the strings that you have up sort of at the top of the neck, if you will, are heavier, thicker, and make lower pitches than the strings that are located more toward the floor or the bottom of the guitar. So the mass and the stiffness of the strings provide for the piano its tuning. That is to say, different physical quantities associated with mass and stiffness of the strings will actually make those strings resonate at different frequencies. And so similarly, your basilar membrane, which is not of uniform stiffness throughout its length, will resonate at different frequencies at different locations. As indicated before, the resonance will be at high frequencies near the base of the cochlea and low frequencies near the apex or the helicotrema. We have to have a trade-off between mass and stiffness, though, right? And so for this vibrating system of the cochlea, the mass is provided by the fluids of the inner ear, the paralymph and the endolymph that I mentioned a moment ago. And the mass of the fluids throughout this organ is essentially a constant. In other words, the mass of the fluids doesn't change from place to place. What changes is the stiffness of the membrane holding the scala media in place, if you will, and separating it from the scala tympani. The basilar membrane is very stiff in this region, and it gets less and less stiff as you wind your way up to the helicotrema. That means that higher frequencies are processed over here and progressively lower and lower frequencies are processed until you make it to the helicotrema, where the lowest of all the frequencies that a human can detect would be audible. So it's a hydraulic system, and the job of the hair cells is to convert hydraulic energy into nerve energy. And I think we've already looked at this. Again, the unique stiffness characteristics, the so-called stiffness gradient of the basilar membrane, is what gives rise to the organization of the membrane with respect to frequency, or its tonotopic organization. Another way to look at it is like this, where we would unroll the cochlea. So here's the scala vestibuli, here's the basilar membrane, and here's the scala tympani. And you can see that high frequency sounds would be processed over here by the round window and the stapes footplate. Progressively lower frequencies will be processed or will produce the most displacement on the basilar membrane further away from the stapes footplate, the oval window, and the round window. And the lowest frequencies will send waves that travel even further away from the stapes footplate and oval window, closer to the helicotrema. So again, different frequencies are processed or produce the most displacement in different physical locations in the cochlea. You can also think of this as a high frequency sound resonating at a location close to the base of the cochlea, whereas a low frequency sound resonates, that is to say produces maximum displacement, resonates at a location closer to the helicotrema or the apex of the cochlea. The next few slides just provide different ways to look at this situation. So we have high frequency sounds over here, 20,000 hertz, progressively lower and lower sounds as we wind our way up toward the apex. Here's an unrolled cochlea, but you see the same idea. The lower in frequency a sound is, the further toward the helicotrema it will produce displacement along the basilar membrane. Kind of all tied together in this image right here. Again, different frequencies resonate at different physical locations along that coiled up structure. We can kind of tie a little bit of this together here and also link it to another interesting idea, which is that of the nerve fibers that make contact with the sensory cells. So over here you see two different frequencies of sound. The blue one is a high frequency sound. How do we know that? Well, look how many cycles there are compared to the number of cycles there are for the other sound, the lower frequency sound. The blue one is cycling through, if you will, repeating that vibration is that sine wave is representing an object that's vibrating more quickly on the blue line than it is on the red line, which is vibrating, but just not the vibration is not repeating itself as many times per second, if you will, or per unit space. And what we see here is an attempt by the artist to have unrolled the cochlea. And so you see this blue wave forms a displacement over here near the base, near the stapes foot plate, etc., near the oval window of the cochlea, whereas the red line produces its maximum displacement over here toward the apex or the helicotrema. Now, I need you to notice how these nerve fibers here, and by the way, you've got 30,000 of them, not 3 dozen or however many are here, but notice how this blue displacement, this blue wave, is going to stimulate fibers only over here, and the red one is going to stimulate fibers only over here. So the idea is that the tuning of the cochlea, the mapping of frequency onto place, right? The mapping of different frequencies onto different physical locations along the cochlea means that different groups of nerve fibers are going to be stimulated when a person hears different sounds. So keep in mind you've got nerve fibers hooked up to this whole thing, right? All through this cochlea, you've got nerve fibers coming off, just like we saw all the way back here. Nerve fibers hooked up to the entire coiled up organ, or here. Nerve fibers hooked up to the whole thing, right? So let's just say for the moment that that blue sound wave is stimulating us in this area, in other words, producing a big wave of fluid displacement over here, and the red one's over here. You can see how different fibers are actually going to be activated by those different sounds, right? And you see it again right here. Different sounds, blue versus red, activate different physical locations in the cochlea and hence different nerve fibers. Again, this organization, this mapping with respect to frequency in the cochlea is called tonotopic organization. And that term refers to the fact that different sounds, different frequencies of sound are going to resonate at different locations on the cochlea. Why? Because the physical structure that's going to be set into motion, the basilar membrane, has this unique stiffness gradient that favors vibration at high frequencies in this area and low frequencies in this area. High frequencies at the base, low frequencies at the apex. What we'll see next time is how the mechanical response of the cochlea gives way to or generates the nerve impulses that will then be responsible for the sensation of hearing. Just testing to see if all of this is working. I then go there, I go back to there, I do this, I do that, etc.